When one thinks of the most dangerous animals on the planet, the mind usually drifts toward the same familiar images: the great white shark moving silently beneath the surface, the crocodile leaping from the shore without warning, or the snake coiled among the fallen leaves of a tropical jungle. Mollusks, by contrast, rarely appear in that mental gallery. They are animals that the collective imagination associates with slowness, with shells on the sand, with soft tentacles that retract at the slightest touch, with the peaceful image of a snail crossing the garden after the rain. That perception, however, is built on a profound ignorance of what this animal phylum is capable of. Mollusks constitute the second most diverse animal phylum on the planet, surpassed only by arthropods. More than 100,000 living species have been described, distributed across virtually all ecosystems, from abyssal trenches to Andean summits, from coral reefs to the freshwater rivers of Central Europe. That colossal diversity conceals survival strategies that go far beyond the protective shell. Venoms that block the nervous system with a precision that the best neurotoxicologists would envy, predators capable of immobilizing and devouring animals several times larger than themselves, parasites that use mollusks as a launching platform to infect millions of human beings every year. All of that exists within this phylum that common sense tends to ignore. The story of dangerous mollusks is also marked by genuine mysteries, mechanisms of action that are still not fully understood, fossil records that suggest behaviors impossible to observe in the present, toxins whose exact composition remains the subject of active research. That component of the unknown is not ornamental, but rather reflects the real state of scientific knowledge about these organisms. We know enough to know that we know almost nothing. The reason why mollusks have been underestimated as a dangerous group has historical and cultural roots worth noting. For much of the 20th century, animal toxicology was organized around venomous vertebrates, snakes, amphibians, fish, and relegated invertebrates to a secondary role in medical and popular literature. Victims of cone snail stings were, for decades, misdiagnosed or treated inadequately because medical personnel in many regions of the Indo-Pacific were unaware of the animal's very existence or underestimated the severity of the envenomation. Only from the 1980s onward, with the growth of tropical marine biology as a discipline and the increase in recreational diving on coral reefs, did emergency medicine begin to systematically incorporate venomous mollusks into its care protocols. That historical delay has consequences that persist. In several of the regions where these animals are most common, healthcare systems still lack sufficient mechanical ventilation equipment to handle multiple cases of severe envenomation simultaneously. The genus Conus comprises around 800 species of marine snails that inhabit primarily the coral reefs of the Indo-Pacific, although some species are distributed across the tropical Atlantic and the Mediterranean. Morphologically, they are snails of remarkable elegance. The shell has a perfect conical shape, hence the name of the genus, and is covered with geometric patterns of a complexity that has fascinated collectors for centuries. That beauty has, in fact, been one of the factors that has historically contributed to accidents with these creatures, because bathers and divers tend to pick them up from the seabed without knowing what they conceal inside. What they conceal is one of the most sophisticated envenomation systems that exist in the animal kingdom. The venom apparatus of the cone snail consists of a venom gland connected to an organ called the radula, which in normal gastropods is a rasping structure used to feed on algae or organic matter. In cone snails, the radula has evolved into a hollow, microscopic, and extraordinarily sharp harpoon, capable of being projected at great speed from an extensible proboscis that can reach any point of the shell, including the end that is normally held in the palm of the hand. That detail is crucial. There is no safe area from which to hold a live cone snail. The venom injected through that harpoon is a mixture of peptides called toxins, and its composition varies depending on the species and, within each species, according to the target prey. Cone snails can feed on polychaete worms, other mollusks, or fish, depending on the species, and the venom is biochemically optimized to act on the nervous system of the corresponding prey group. Piscivorous species, that is, those that hunt fish, are the most dangerous to humans because their venom acts on ion channels that are highly conserved evolutionarily among vertebrates, which means that the mechanisms that kill a small fish also work in the nervous system of an adult human being. Toxins act by selectively blocking different types of ion channels, voltage-dependent sodium, calcium, and potassium channels, as well as nicotinic acetylcholine receptors. This multiple and simultaneous selectivity produces a neuromuscular paralysis that can progress within minutes to complete respiratory failure. The first symptoms include extreme immediate pain at the injection site, followed by numbness that spreads from the site of the sting to the rest of the body, double vision, difficulty swallowing, and progressive muscular weakness that eventually prevents autonomous breathing. There is no specific antidote for any species of conus. Treatment is exclusively supportive, based on mechanical ventilation, until the body metabolizes the venom. The most dangerous species and the one with the highest number of documented human deaths is Conus geographus, popularly known as the geography cone snail because of the pattern of its shell, which resembles a map with irregular islands on an orange background. This species can reach 15 centimeters in length, inhabits coral reefs between 1 and 20 meters deep, and is actively nocturnal. During the day, it remains buried in the sand or hidden under corals. So, divers who handle it are rarely aware that it is alive. It is estimated that the mortality rate without medical treatment in cases of envenomation by Conus geographus exceeds 70%, which makes it one of the most lethal invertebrates in the world in terms of proportional mortality from a sting. The estimated lethal dose of its venom in humans is on the order of micrograms per kilogram of body weight, which implies that the amount injected in a single sting is sufficient to kill several adults. Historical cases of envenomation describe a clinical progression that can range from first contact to death in as little as 4 hours, although in most cases the time window is longer, between 12 and 24 hours, which in theory would allow medical intervention if the patient is near adequate facilities. The problem is that accidents usually occur in remote areas of the Indo-Pacific, with limited access to mechanical ventilation. 2 other representatives of the genus deserve specific mention. Conus textile, the textile cone snail, is the second most dangerous species of the genus and presents a relevant behavioral peculiarity. It is significantly more active during the day than Conus geographus and tends to bury itself less deeply in the substrate, which increases the likelihood of accidental contact with divers. Its shell pattern, a network of brown triangles on a cream background that indeed resembles fabric, makes it one of the most visually attractive in the genus, and therefore one of the most frequently collected by collectors. Conus striatus, the striated cone, completes the trio of species with the highest documented risk. Its stings have been reported in Polynesia, the Philippines, and Indonesia, and although the documented mortality is lower than that of Conus geographus, the speed of symptom progression, in some cases, is greater. The combination of striking shell, relatively active behavior during diving hours, and highly potent venom makes these 3 representatives of the genus the most likely candidates for the next serious undocumented accident on some Pacific reef. Paradoxically, the same molecular precision that makes conotoxins lethal has made them one of the most promising sources of new drugs of the 21st century. The U.S. FDA approved in 2004 ziconotide, a drug derived from a conotoxin of Conus magus, as a last resort analgesic for severe chronic pain resistant to morphine, Ziconotide acts by blocking a specific subtype of neuronal calcium channel and produces analgesia without the addictive effects of opioids. Since then, thousands of different peptides have been identified in the venom of different conus species, and active pharmacological research is seeking candidates to treat epilepsy, cardiac arrhythmias, Parkinson's disease, and Alzheimer's disease. The phylum Mollusca, through this genus, has contributed to modern medicine in a way that few imagine. Octopuses are creatures of recognized and documented intelligence. They solve mazes, use tools, learn by observation, and have short and long-term memories. They are also, for the most part, completely harmless to humans despite possessing a chitinous beak capable of piercing the skin. Within the order Octopoda, however, there exists the genus Hapalochlaena, which groups 4 species of small octopuses distributed along the coasts of the Indo-Pacific, from Japan to Australia, and which represent the only genuinely dangerous exception to that rule of harmlessness. Hapalochlaena lunulata, H. maculosa, H. fasciata, and H. neostracea are small octopuses that rarely exceed 12 centimeters in diameter, including the tentacles, and which at rest have a brown or yellowish coloration that makes them practically invisible against rocks and sandy bottoms. That discretion disappears when the animal feels threatened. In tenths of a second, the body is covered with rings and spots of a bright iridescent blue, produced by the reflection of light in chromatophores and iridophore cells, a chromatic warning that has evolved as a signal of danger to predators and that biologists call aposematism. The venom of the blue-ringed octopus is not produced by the animal itself, but by symbiotic bacteria of the genus Pseudoalteromonas that live in its salivary gland. These bacteria synthesize tetrodotoxin, also known as TTX, one of the most potent non-protein natural toxins known. Tetrodotoxin acts by specifically blocking voltage-gated sodium channels in the membranes of nerve and muscle cells, preventing the propagation of the action potential and producing a neuromuscular paralysis that can affect the heart muscle and the diaphragm. The lethal dose in humans is estimated at around 2 micrograms per kilogram of body weight. A single bite from a blue-ringed octopus can contain enough dose to kill 26 adults. The major clinical problem of tetrodotoxin poisoning is that it does not produce pain. The bite of the blue-ringed octopus can go unnoticed because the beak is small and the venom does not generate immediate local irritation. Many victims do not realize they have been bitten until neurological symptoms begin to appear, which include numbness of the lips and tongue, nausea, difficulty speaking and swallowing, and an ascending paralysis that can lead to respiratory arrest in 15 minutes. Without artificial ventilation, death by asphyxia can occur within 1 hour of the bite. As with conotoxins, there is no specific antidote. The only treatment is sustained mechanical ventilation until the toxin is excreted, a process that can require between 18 hours and several days. The presence of tetrodotoxin in marine animals is not limited to the genus Hapalochlaena. The same toxin, produced by symbiotic or commensal bacteria, has been found in the pufferfish or fugu of the families Tetraodontidae and Diodontidae, which constitutes one of the major food safety problems in Japan and South Korea, in certain species of salamanders of the genus Taricha in California, in frogs of the genus Atelopus in South America, and in several other mollusks, including certain gastropods of the genus Nasarius. This very broad phylogenetic distribution suggests that tetrodotoxin was not independently invented many times, but that there exists a network of bacterial transfer through marine food chains whose complexity has not yet been fully mapped. The mystery of how these bacteria are distributed among such evolutionarily distinct hosts remains the subject of active research. For centuries, sailors from all coastal cultures of the world coincided in describing marine creatures of enormous size that attacked ships and dragged men to the bottom of the ocean. In Norse mythology, they were the kraken. In the chronicles of Portuguese navigators of the 16th century, they appear as sea serpents of impossible proportions. In the accounts of Jules Verne, they found their most well-known literary expression. Behind all those narratives, there was a real animal, Architeuthis dux, the giant squid, whose existence was definitively accepted by science in the 19th century, when corpses or fragments began to appear on beaches around the world. Architeuthis dux is the largest invertebrate on the planet in length, with documented individuals reaching 13 meters from the tip of the tentacles to the end of the mantle. The eyes of the giant squid are the largest in the animal kingdom, with diameters that can exceed 30 centimeters, an adaptation to the dimness of the depths where it lives, between 200 and 1,000 meters deep, in all the oceans of the world. It is an active predator that feeds on fish and other cephalopods, and is in turn the main prey of the sperm whale. Whales of that species frequently appear with circular scars produced by the suckers of the giant squid, suckers that are equipped with serrated edges of hard chitin that function like saws when rotated. For more than a century and a half after its existence was accepted by science, Architeuthis was known only through corpses or fragments, turning the animal into a ghost species whose living biology was completely unknown. That situation changed in September 2004, when Japanese researchers from the National Institute of Polar Research captured a photographic sequence of a live specimen at 800 meters depth in the Pacific Ocean, east of the Ogasawara Islands. The images showed the squid actively attacking the bait of the trap, with its tentacles extended in the form of a crown, a behavior that contradicted the dominant hypothesis that it was a slow-moving predator that waited for its prey rather than pursuing it. In 2012, the same institution obtained the first high-definition video images of an adult specimen in its natural habitat, and the sequences confirmed that the animal is capable of rapid and coordinated movements that contradict decades of assumptions based on the morphology of corpses. The question of whether Architeuthis represents a threat to humans is more complex than it might appear at first glance. There are no verified records of fatal attacks on divers or swimmers, which has a relatively simple explanation. The giant squid inhabits depths that humans do not normally access without submarines. However, there are historical accounts of shipwrecks during the Second World War, in which survivors on rafts described attacks by large cephalopods that dragged men into the sea during the night. These testimonies have not been scientifically verified, and the debate over whether they were attacks by Architeuthis, by other species of large squid, or by phenomena misinterpreted by survivors in extreme states of stress and delirium due to hypothermia, remains open. If Architeuthis is the longest squid, Mesonicoteuthis hamiltoni, known as the colossal squid, is the most massive in terms of weight, with estimates suggesting that mature adults may weigh between 400 and 700 kilograms. It lives in the depths of the Antarctic Ocean, often below 2,000 meters, and is even more unknown than the giant squid. The number of complete specimens examined by science does not exceed a dozen, most of them accidentally captured in the stomachs of sperm whales or in long-line fishing nets of Antarctic vessels. What distinguishes Mesonicoteuthis from the rest of known squids is not only its size, but its armament. Its tentacles are equipped with rotating hooks of chitin, not simple suckers, which can rotate on their axis and which researchers describe as capable of producing serious wounds in any soft tissue they catch. In addition, it possesses the largest beak, in proportion to the body, of any known squid, with chitinous jaws capable of generating extraordinary bite pressures. The reproductive biology, hunting behavior, movement patterns, and lifespan of this species are practically unknown, because a live specimen has never been observed in its natural habitat under conditions that would allow systematic documentation. Everything that is known comes from fragmented corpses and from inferences based on the marks it leaves on its prey. There exists a category of danger associated with mollusks that does not involve venom or direct predation, but something more subtle and in many ways more devastating on an epidemiological scale. The role of intermediate host in the life cycles of parasites that massively affect human health. This role turns certain species of snails into one of the most significant factors of morbidity and mortality in the world, although that relevance is almost invisible to the general public. The biological basis of the phenomenon lies in the fact that many trematodes, that is, parasitic flatworms of the Digenean group, obligatorily require passing through an asexual reproduction phase within a gastropod mollusk before they can infect their definitive host, which in many cases is a vertebrate, including humans. Inside the snail, the parasite multiplies exponentially, producing thousands of larvae called cercariae, which are released into the water and actively seek the next host. This mechanism of parasitic amplification within the mollusk means that a single infection of the snail can result in the release of tens of thousands of infective larvae into the aquatic environment, transforming any body of water that harbors populations of these snails into a permanent health risk. Schistosomiasis, also called bilharzia, is a parasitic disease caused by trematodes of the genus Schistosoma and constitutes the second parasitic disease with the greatest impact on public health worldwide, second only to malaria. According to data from the World Health Organization, it affects more than 200 million people in 78 countries, mainly in sub-Saharan Africa, although also in parts of Asia and Latin America, and causes between 200,000 and 300,000 deaths annually directly, with a burden of chronic disease estimated in millions of years of life with disability. The intermediate hosts of the different schistosoma are freshwater snails of the genera Biomphalaria, for S. mansoni, Bolinus, for S. hematobium, and S. intercalatum, and Oncomelania, for S. japonicum. These snails are small, measuring from less than 1 centimeter to 3 centimeters in diameter depending on the species, and inhabit slow surface waters, rice fields, irrigation channels, ponds, and the edges of rivers and lakes. Human infection occurs when the cercariae, released by the snail, actively penetrate through intact skin during contact with the water, without the need for ingestion or any other mechanism. It is enough to walk or swim in infested water for a few minutes for the larvae to manage to cross the epidermis. Once inside the human body, the cercariae mature into adults that settle in the blood vessels of the intestine or the urinary bladder, depending on the species, where they can live between 3 and 30 years producing eggs continuously. These eggs are responsible for the pathological damage. Some are expelled with feces or urine, closing the cycle upon reaching fresh water, where they hatch and seek a snail. But a significant fraction becomes trapped in the host's tissues, mainly in the liver, spleen, and intestine, where they provoke a chronic granulomatous inflammatory response that produces progressive fibrosis, portal hypertension, severe anemia, and, in the case of S. hematobium, a documented increase in the risk of bladder carcinoma. The complexity of this parasitic cycle, which involves species specificity both in the mollusk and in the definitive host, makes the control of schistosomiasis one of the most challenging problems in contemporary tropical medicine. Attempts to control the disease by attacking the most vulnerable link in the cycle, the snail host itself, have had a long and uneven history. In the first half of the 20th century, several African and Latin American countries implemented massive campaigns to eliminate Biomphalaria and Bolinus using chemical molluscicides, mainly niclosamide, which proved effective in the short term but generated devastating side effects on non-target aquatic invertebrate communities, including beneficial insects and other species of mollusks that naturally compete with the parasite's hosts. The interruption of these campaigns was invariably followed by a rapid recovery of snail populations and a resurgence of transmission. At present, the dominant strategy of the WHO is based on mass treatment with praziquantel, the only effective drug against all schistosoma, but without simultaneously addressing the reduction of contact with water or the elimination of the intermediate host. Pharmacological treatment only reduces morbidity without interrupting transmission, which implies that the same communities are retreated year after year in a cycle that the global health system has been unable to break for decades. Fascioliasis is a parasitic disease caused by Fasciola hepatica, the liver fluke, a trematode whose intermediate host is the freshwater snail Galba truncatula, a species with an almost cosmopolitan distribution that inhabits the mud of stream edges, ponds, and wet areas. Fascioliasis mainly affects cattle and sheep, causing enormous economic losses in global livestock farming, but it also constitutes a zoonosis with significant human impact. The WHO estimates that between 2 and 17 million people are infected worldwide, with another 91 million at risk, especially in Bolivia, Peru, Ecuador, Egypt, and Iran. Human infection occurs almost exclusively through the ingestion of Metasacariae attached to aquatic plants such as watercress, on which the encysted larvae survive while the plant is fresh, and which humans consume raw in salads. The larvae released in the intestine perforate the intestinal wall, migrate through the abdominal cavity, penetrate the liver, and travel through the hepatic parenchyma for weeks before finally settling in the bile ducts, where they can live between 10 and 20 years. The acute phase of the disease, while the larvae migrate through the liver, produces high fever, intense abdominal pain, and marked eosinophilia. The chronic phase is characterized by recurrent biliary inflammation, duct obstruction, and a high risk of bacterial cholangitis. The standard treatment is triclobendazole, a drug for which resistance is beginning to be documented in some parasite populations. The nautilus, which includes the genus Nautilus and the recently separated Allonautilus, is one of the oldest animals on the planet in terms of body plan. Fossils of nautiloids date back 500 million years, and current individuals are morphologically so similar to their Cambrian ancestors that they are colloquially classified as living fossils. They inhabit the Indo-Pacific at depths ranging from 100 to 600 meters during the day, ascending to shallower waters at night to feed. A pattern of vertical migration they achieve using gas in the chambers of their shell, controlling their buoyancy through a sophisticated system of sealed chambers connected by a siphuncle. The nautilus is an active predator of crustaceans, mainly hermit crabs and small lobsters, which it captures with its tentacles, which can number between 80 and 90 in adults and lack suckers, using instead adhesive structures coated with mucus. The chitinous beak of the nautilus is capable of breaking the exoskeletons of its prey with ease. For humans, the nautilus does not represent any direct danger. Its dentition cannot pierce human skin with the bite force it generates, and it produces no known venom. However, its inclusion in this account is due to its position in the food chain and the complexity of its hunting system, which disproves the idea that externally shelled mollusks are passive, filter-feeding animals. Polyplacophorans, popularly known as chitons, are mollusks that many zoologists consider to have a primitive design. The body is covered by 8 articulated plates of calcium carbonate, giving them the appearance of a small marine armadillo, and they move slowly over rocks and hard surfaces, scraping algae with a radula reinforced with magnetite, the hardest material biologically produced by any known animal. This radula can withstand pressures of several gigapascals, which has sparked enormous interest in materials engineering. What is surprising from the point of view of danger is that at least one species of chiton has developed active carnivorous behavior. Placiforella velata, which inhabits the coasts of the North American Pacific, has been observed raising the anterior end of its body to create a trap, so that when small crustaceans or worms pass underneath, the animal captures them by crushing them with the cephalic shield before scraping them with the radula. This behavior, documented in the laboratory and confirmed in the field, represents an evolutionary transition that turns an animal considered herbivorous into an active predator, albeit of very small prey. The exact mechanism by which Placiforella detects its prey, whether through chemoreceptors, vibration, or tactile stimulation, remains not fully elucidated. Bivalves, which include mussels, clams, oysters, scallops, and cockles, are filter feeders that obtain their food by pumping large volumes of water through their gills and retaining suspended organic particles, including phytoplankton, bacteria, and detritus. This extraordinarily efficient feeding strategy has a major toxicological consequence when the water they filter contains toxin-producing microalgae. These toxins accumulate in the tissues of the bivalve at concentrations that can exceed those present in the water by thousands of times. The bivalve, which has resistance mechanisms against these toxins, remains apparently healthy while accumulating doses that are lethal to the vertebrates that consume it. Blooms of toxic microalgae, called red tides, although they do not always involve that color, are natural phenomena whose documented frequency and intensity have increased in recent decades, probably in relation to the enrichment of coastal waters with nutrients from agricultural and urban effluents, and with changes in sea surface temperature associated with climate warming. Dinoflagellates of the genus Alexandrium produce saxitoxins, responsible for paralytic shellfish poisoning. Karenia brevis produces brevetoxins, associated with neurotoxic shellfish poisoning. Pseudonitschia produces domoic acid, which causes amnesic shellfish poisoning. These three syndromes have in common that bivalves act as passive vectors of the toxins to human consumers. Saxitoxin and its analogues are blockers of voltage-gated sodium channels with a potency comparable to tetrodotoxin and constitute the cause of paralytic shellfish poisoning, known by its acronym PSP. Symptoms begin between 30 minutes and 2 hours after the consumption of contaminated shellfish and progress from tingling and numbness of the lips, tongue, and face to generalized muscular paralysis that may include the respiratory muscles. In severe cases, death from respiratory failure can occur between 2 and 12 hours after ingestion, and the mortality rate in situations without access to mechanical ventilation has historically been estimated between 8 and 17% of symptomatic cases. There is no specific antidote. Saxitoxin has a history that goes beyond marine toxicology. It was identified by biological weapons research programs during the Cold War as a possible chemical warfare agent, and it was included in the 1993 Chemical Weapons Convention. For decades, the CIA maintained reserves of saxitoxin in classified facilities. In 1975, the director of the CIA admitted before Congress that the agency had retained 11 grams of the toxin, in violation of presidential orders to destroy biological warfare arsenals, which illustrates the extent to which the chemistry of mollusks can have implications that transcend marine ecosystems. Domoic acid, produced mainly by diatoms of the genus Pseudonitschia, causes amnesic shellfish poisoning, a syndrome whose existence was not recognized until 1987, when, in the Canadian province of Prince Edward Island, 4 people died, and more than 150 became seriously ill after consuming mussels cultivated in the waters of the Gulf of St. Lawrence. Epidemiological investigation revealed that the mussel had accumulated domoic acid produced by a bloom of Pseudonitschia that had gone unnoticed. Symptoms include acute gastrointestinal problems followed, in the most severe cases, by confusion, disorientation, loss of short-term memory, seizures, and coma. Autopsies of the deceased showed selective necrosis of the hippocampus, the central brain structure for the formation of new memories, caused by excessive activation of glutamate receptors by domoic acid. Several of the survivors who suffered severe forms of the syndrome presented permanent neurological memory deficits. Pomacea canaliculata, known as the apple snail due to the yellow-orange color of many of its individuals, is a freshwater gastropod native to the river basins of the Paraná and Uruguay in South America, which has been intentionally spread around the world as a food source for aquaculture and as an aquarium ornament. This spread has resulted in one of the most documented and problematic biological invasions of the last 50 years. The species is currently established in more than 30 countries in Asia, Africa, Europe, and America, where it displaces native invertebrate fauna, destroys rice crops and aquatic plants, and acts as an intermediate host of a parasitic nematode with serious neurological consequences. The nematode in question is Angiostrongylus cantonensis, the rat lungworm, whose normal life cycle involves rodents as definitive hosts and terrestrial or aquatic mollusks as intermediate hosts. Humans are accidental hosts who become infected by consuming raw or undercooked mollusks or vegetables contaminated with mucus from infected mollusks, or water that has been in contact with parasitized mollusks. In the human host, the larvae of the nematode fail to complete their normal cycle and instead migrate to the central nervous system, where they cause eosinophilic meningoencephalitis that can produce paralysis, permanent sensory deficits, and in the most severe cases, death. Angiostrongylliasis, caused by Angiostrongylus cantonensis, was first described in Canton, China, in 1945, and for decades it was considered a disease mainly of the Indo-Pacific region. The global expansion of Pomacea, canaliculata, and other host mollusks has radically altered that geographic distribution. Autochthonous cases have been documented in Hawaii, in the Caribbean, in sub-Saharan Africa, and in southern Europe. Most cases are mild and resolve spontaneously within a few weeks, but the fraction of severe cases, which involve extensive larval migration in nervous tissue, produces a clinical picture that can include anything from severe headache and neck stiffness to cranial nerve paralysis, blindness, hydrocephalus, and death. One of the most disturbing aspects of this disease is that treatment with antiparasitics can in some cases worsen the clinical picture, because the massive death of larvae within the central nervous system triggers an inflammatory response that can be more damaging than the live parasites themselves. Current therapeutic protocols combine corticosteroids to control inflammation with antiparasitics, such as albendazole in selected cases, but the uncertainty about the optimal regimen reflects the fact that there are no randomized clinical trials of sufficient quality to establish evidence-based recommendations. The reason is simple. The disease mainly affects rural populations in low-income countries, where investment in clinical research is limited. The Humboldt squid, Dosidicus gigas, is the largest cephalopod in the eastern Pacific, with adult individuals reaching 2 meters in total length, including tentacles and weights exceeding 50 kilograms. It inhabits the Humboldt Current, the cold ocean current that runs along the southeastern Pacific from Chile to Mexico, and is one of the most important vertebrate and invertebrate predators in the pelagic ecosystem of that region, consuming fish, crustaceans, and other cephalopods in enormous quantities. Its common name on the coasts of Mexico, where it is the target of an intense artisanal fishery, is red devil, a designation that is not arbitrary. Unlike the giant or colossal squid, which live at depths inaccessible to most humans, Dosidicus gigas performs nocturnal vertical migrations that bring it to surface waters between 0 and 200 meters deep, where it actively feeds in groups that can number in the thousands of individuals. These aggregations are the context in which the most documented incidents with this species have occurred. Lobster diving fishermen on the coast of Baja California have described episodes in which the squids, apparently stimulated by movement, and by the presence of blood from captured fish, surrounded divers in large groups and began to pull with their suckers, armed with chitinous hooks, and to bite with the beak. What distinguishes Dosidicus gigas from other squids known as a risk to humans is not only size or armament, but the combination of aggressiveness, gregarious behavior, and distribution in accessible waters. The suckers of Dosidicus gigas, unlike those of other squids, are equipped with toothed rings and in some cases with movable hooks that significantly increase their ability to grasp prey. When a group of Humboldt squids attacks a large fish or an object they perceive as prey, the coordination of the group produces a pulling effect in multiple directions simultaneously that can literally tear the prey into fragments. Divers who have experienced attacks by these squids describe lacerations in the skin caused by the hooks of the suckers, extensive bruising, and attempts by the animal to drag the diver downward, which in several cases has resulted in decompression accidents when resisting the attack by ascending too quickly. Among the most documented episodes is the one experienced by several commercial fishing divers off the coasts of Baja California Sur in the winter of 2002, when groups of Humboldt squids, estimated in the hundreds of individuals, attacked divers working at depths between 10 and 20 meters at night. One of the divers was dragged downward out of the sight range of his companions before managing to free himself. Others suffered tears of several centimeters in their arms and legs. These testimonies, collected by journalists and in later years by researchers from the Monterey Bay Aquarium Research Institute, became the empirical basis on which the scientific debate was built regarding whether attacks by Dosidicus gigas on humans have a predatory motivation or are simply a form of tactile exploration amplified by group behavior. Neither of the two hypotheses has been definitively refuted, and the scientific literature treats them as complementary rather than mutually exclusive. The question of whether these attacks are genuine hunting behavior directed at humans or simply a response of exploration and manipulation of unusual objects in the environment is debated among researchers. What is documented is that the behavior of Dosidicus gigas changes dramatically in the presence of blood or other chemical stimuli associated with injured prey. Under those circumstances, the threshold of aggressiveness drops notably, and the squid become difficult to repel. Artisanal fishermen from Baja California who work with nets at night are well aware of this phenomenon and use red light flashlights to confuse the squid, as it has been observed that reddish colors have a calming effect on them, possibly because the spectral range of red light penetrates less underwater and the squid associate it with lower visibility. One of the most concerning phenomena in the ecology of Dosidicus gigas is that in the last 2 decades, its distribution range has expanded notably northward, with individuals regularly appearing along the coasts of California, Oregon, and Washington, where historically the species was rare or absent. This expansion has been associated with Pacific climate variability and possibly with the warming of surface waters, although the exact causal relationship has not been established. In parallel, the overfishing of its competitors and predators, especially hake and tuna, has reduced pressure on its populations and allowed substantial demographic growth. The result is that a species that until a few years ago was problematic mainly for fishermen in Mexico and Peru is becoming a permanent element of the North American Pacific, with implications for local ecosystems and for the safety of recreational divers that have not yet been exhaustively evaluated. The zebra mussel, Dreissena polymorpha, is a bivalve native to the Caspian Sea and Black Sea basins that was accidentally introduced into the Great Lakes of North America in the late 1980s, probably through ballast water from cargo ships arriving from Eastern Europe. In the following decades, it spread through virtually the entire hydrographic network of the eastern United States and Canada, becoming the textbook example of a devastating biological invasion. What makes this animal relevant in the context of danger is not direct harm to humans, but the scale and speed of its impact on the freshwater ecosystems it colonizes, and the cascading consequences that impact generates. Dreissena polymorpha can reach densities of up to 700,000 individuals per square meter on lake and river bottoms, a biomass that turns any hard surface into a living carpet of filter feeders. At those densities, the filtration capacity of the collective population often exceeds the total volume of the lake in cycles of days, meaning they literally remove from the water all suspended organic matter, including phytoplankton, at a rate that completely surpasses natural replenishment. The result is an extreme clarification of the water that appears positive at first glance, but actually represents a catastrophic disruption of the planktonic trophic web. Without phytoplankton, zooplankton collapse. The fish larvae that depend on it collapse, and subsequently adult fish, aquatic birds, and the mammals that feed on them. The impact of the zebra mussel on human health has two main vectors. The first is the proliferation of toxigenic cyanobacteria. By removing eukaryotic phytoplankton from the water column, zebra mussels paradoxically favor cyanobacteria, which produce cyanotoxins such as microcystins and anotoxins, and which the mussels actively reject while filtering, accumulating them in much lower proportion than normal phytoplankton. Cyanobacterial blooms in lakes invaded by Dreissena have increased in frequency and intensity, producing beach closures, contamination of drinking water sources, and episodes of poisoning in domestic animals. The second vector is the amplification of Vibrio cholerae. Laboratory studies have shown that Dreissena concentrates this bacterium in its tissues, and that its presence in drinking water systems can hinder conventional purification processes. The giant African snail, Achatina fulica, is the largest land snail in the world, with adults that can reach 20 centimeters in length and weigh up to 300 grams. Native to East Africa, it was transported as a food source, as a pet, and for use in the cosmetic industry to virtually all tropical and subtropical continents during the 20th century, becoming a major agricultural pest in Asia, the Pacific, the Caribbean, and parts of South America. In Hawaii, where it was introduced in 1936, the fight against its spread has cost tens of millions of dollars and has involved the introduction of biological predators whose own ecological impacts have, in some cases, proven more damaging than the original pest. The relevance of Achatina fulica to human health goes beyond agricultural damage. This snail acts as an intermediate host of Angiostrongylus cantonensis, the nematode that causes eosinophilic meningoencephalitis, described in the previous chapter, and also of Angiostrongylus costarricensis, a related species that produces abdominal angiostrongyliasis, characterized by inflammation of the large intestine and the appendix. The parasitic load of Achatina fulica populations in different regions varies significantly, and in some areas of the Caribbean and the Pacific, infection rates with A. cantonensis exceeding 80% of sampled individuals have been found, making any contact with the animal, even indirect through vegetables or surfaces it has passed over, a potential risk of infection. An epidemiologically important aspect of Achatina fulica is that its size makes it particularly attractive to children, who tend to handle it directly, and to communities that consume it as a protein source. Several outbreaks of eosinophilic meningoencephalitis in California, Brazil, and Pacific Islands have been attributed to handling these snails without gloves or to consuming vegetables contaminated with their mucus. The snail's mucus may contain third-stage larvae of A. cantonensis that are infectious orally or through skin abrasions. Public health campaigns in areas invaded by A. fulica routinely include the recommendation not to handle the snail with bare hands and to carefully wash any vegetables from gardens or orchards where the snail is present. Thomoctopus mimicus, the mimic octopus, was first scientifically described in 1998 from specimens found on the sandy bottom of the Lombok Strait in Indonesia, although it has since been documented in other locations in Southeast Asia. It is a medium-sized octopus, rarely exceeding 60 centimeters in total length, with a beige base coloration with brown bands, and it has developed an unprecedented capacity for behavioral mimicry documented in the animal kingdom. It does not limit itself to changing skin texture or color to camouflage with the background, as many cephalopods do, but actively imitates the form and behavior of other extremely dangerous animals in the same environment. Documented imitations include the stonefish, one of the most venomous fish in the world, which Thaumoctopus mimicus replicates by flattening its body against the bottom with its tentacles extended in the characteristic position of that fish. The venomous sole, which the octopus imitates by burying 6 of its tentacles in the sand and undulating the remaining 2 with the fish's wave-like motion while swimming near the bottom, and the rock-dwelling stonefish, whose rough texture and motionless posture the octopus reproduces with remarkable fidelity. The question of whether these imitations are selected contextually, that is, whether the octopus chooses which animal to imitate depending on the specific predator threatening it, has not been definitively resolved, although field observations suggest that there is at least some degree of adaptive selection. Although the mimicry of Thaumoctopus mimicus is primarily a defensive strategy against predators, it also has implications for its predatory behavior. By imitating animals that are harmless or uninteresting to potential prey, the octopus can approach crustaceans and small fish without triggering their escape response. This dual use of mimicry, as a shield against predators and as a trap against prey, places Thaumoctopus mimicus in a position of high-efficiency predator within the sandy bottom ecosystems of Southeast Asia. Its venom, produced in the posterior salivary gland, contains a mixture of paralyzing toxins whose exact composition has not been fully characterized, although it is known to be effective in immobilizing the crustaceans that make up most of its diet. Diuretic shellfish poisoning, known by the acronym DSP from its English name, is the most frequent intoxication from the consumption of contaminated bivalves worldwide, although it is also the least lethal compared to paralytic or amnesic poisoning. It is caused mainly by octadeic acid and its analogues, the dinophysis toxins, produced by dinoflagellates of the genus Dinophysis and by Prorocentrum lima. Bivalves that filter these microalgae accumulate the toxins in their digestive gland without apparent harm, and the toxins remain active in the tissues for weeks after the microalgal bloom episode has ended, which greatly complicates monitoring systems and the preventive closure of harvesting areas. The symptoms of diuretic poisoning are gastrointestinal, nausea, vomiting, profuse diarrhea, abdominal cramps, and chills that begin between 30 minutes and 12 hours after consumption, and resolve spontaneously within a few days in most cases. Direct mortality is practically null, but octadeic acid is a potent inhibitor of protein phosphatases, enzymes that regulate cell proliferation, and epidemiological studies in populations with chronic exposure to low doses of these toxins have found associations with increased risk of colorectal cancer, although causality has not been definitively established. This potential carcinogenic aspect adds a long-term risk dimension that goes beyond the acute episode of gastroenteritis. The current state of knowledge on mollusk toxicology is marked by significant gaps that are not simply academic technicalities, but questions with direct practical consequences for emergency medicine and public health. One of the most striking concerns the cone snails themselves. Although conotoxins as a chemical class are relatively well characterized, each species produces an idiosyncratic cocktail of peptides that may contain between 100 and 200 distinct components. And for most of the less studied species, even the basic toxin profile has not been determined. This means that in a case of envenomation by a little-known species, physicians treat empirically without being able to predict which components of the venom are responsible for the clinical picture, nor what its temporal progression will be. Another significant mystery concerns the toxins of octopuses other than the blue-ringed ones. Several species of octopuses have been reported in the medical literature as responsible for bites with neurological or systemic effects that go beyond what would be expected from a simple wound caused by the beak. But the responsible agent has not been identified in most of those cases. Some researchers postulate the existence of additional toxins in the saliva of those octopuses produced by symbiotic bacteria different from those that produce tetrodotoxin, but the systematic chemical characterization of the saliva of the approximately 300 known species of octopuses has barely begun. In that vast space of the unexplored, the possibility of finding new venoms with pharmacological properties of interest is genuinely high. The family Conidae is not the only one that has developed venom injection systems among carnivorous gastropods. The Terebras, gastropods of the family Terebridae that inhabit the same reefs as the cones and that have much more elongated spiral shells, also produce conotoxins or similar compounds in the salivary gland and possess a modified radula capable of injecting venom, although their potency is considerably lower and no human deaths attributable to them have been documented. The Turritella and the augurs, vernacular names of several species of Terebridae, are frequently handled without caution by divers who confuse them with empty shells, and although the risk of death is low, the stings produce intense, localized pain and in some cases mild, systemic reactions. More enigmatic is the case of the genus Colubraria, a family of small predatory gastropods that in recent years has been the subject of research due to the presence of bioactive compounds in its salivary gland that function as anticoagulants when the animal feeds on sleeping fish. Colubraria reticulata and related species have a feeding behavior that researchers describe as parasitic. They attach to the body surface of fish during the night, when these are in a phase of inactivity, and suck blood for several hours by injecting anticoagulant saliva, without the fish seeming to wake up or react. The chemical composition of that anticoagulant saliva and its potential interaction with coagulation in other vertebrates, including humans, is being actively studied. The first results suggest the presence of compounds analogous to hirudin from leeches, which makes them potential candidates for the development of new generation antithrombotics. An additional complication in assessing the danger posed by mollusks is the problem of cryptic taxonomy, the existence of multiple biologically distinct species that are morphologically indistinguishable or almost indistinguishable, and that can only be differentiated through genetic analysis. In the genus Conus, molecular studies have revealed that what for decades was considered a single species actually comprises several independent evolutionary lineages with potentially different venom profiles. For collectors, divers, and fishermen who handle these animals, the impossibility of visually identifying the exact species they have in hand implies that risk assessment, based on the visual appearance of the shell, is inherently imprecise. Similarly, in the genus Hapaloclaena, the identification of juvenile individuals, which have the same venomous capacity as adults but whose pattern of blue rings is less conspicuous, requires a level of field taxonomy expertise that most bathers do not possess. The taxonomic revision of the genus Conus, using molecular phylogeny tools, has led to a profound reorganization of the nomenclature. Several researchers propose fragmenting the genus into more than 80 distinct genera based on genetic differences and on the diversity of the venom apparatus. Although this proposal is controversial, and many specialists prefer to maintain the traditional denomination using subgenera, this taxonomic dispute has direct practical consequences. If the fragmentation is accepted, the same animal may have different names in different medical and biological databases, which complicates communication between clinicians from different countries in the event of an envenomation case. The question of what level of genetic difference justifies separation into distinct genera in such a diverse group and with such extraordinary venom, diversity has no agreed answer, and meetings of taxonomists specialized in Conidae continue without producing a definitive agreement. The fossil record of mollusks, exceptionally complete compared to that of other invertebrates thanks to the durability of shells, raises questions about past behaviors that have no observable modern equivalent. Ammonites, extinct cephalopods that dominated the Mesozoic seas for 180 million years, and that in some genera reach sizes exceeding 2 meters in diameter, are the most spectacular example. The reconstruction of their biology, based on their closest living relatives, the nautiluses, suggests that some large-sized genera would have been top predators of Jurassic and Cretaceous marine ecosystems, capable of attacking prey of considerable size. However, direct evidence of hunting behavior in the fossil record is fragmentary, and discussions about whether ammonites were active predators, opportunistic scavengers, or filter feeders are far from settled. A paleontological mystery related to mollusks that has generated considerable debate is that of the so-called Monte Cristo monster, a series of Triassic remains found in Nevada and described in 2011 by the paleontologist Mark McMenamin. McMenamin interpreted the arrangement of ichthyosaur vertebrae at the site as evidence that a giant cephalopod, comparable in size to a modern sperm whale, had hunted and reorganized the remains of its prey into a deliberate pattern, a hypothesis that generated both media fascination and an extraordinarily skeptical reception from the paleontological community. Most experts consider that the observed pattern can be explained by currents or by the natural decomposition of carcasses without the need to invoke a cephalopod predator of monstrous size. And the hypothesis remains without definitive confirmation or refutation. What the episode reveals, beyond its plausibility, is that Mesozoic cephalopods reach diversities and sizes extreme enough that speculation about their predatory capacities is at least biologically plausible. Within the group of modern mollusks, there are behaviors known from a single observation or from such a small number of records that it is impossible to know whether they are common behaviors or exceptional events. The trapping behavior of Placiforella velata was first observed in the laboratory in the 1970s and only recently confirmed under natural conditions. The possibility that similar behaviors exist in poorly studied mollusk species is not speculative, but a reasonable inference from the known distribution of behavioral complexity within the phylum. The deep Pacific seabeds, the less explored reefs of the Indian Ocean, and the oxygen minimum zones that surround some ocean currents harbor communities of mollusks whose basic biology is unknown. The review of the most dangerous mollusks in the world reveals that danger within this phylum does not have a single form, nor does it emerge from a single evolutionary mechanism. It is distributed throughout the entire taxonomic diversity of the group, expressed in molecularly precise toxins in cone snails and blue-ringed octopuses, in passively accumulated paralytic venoms in bivalves, in the size and mechanical armament of giant and colossal squids, in the active predatory behavior of the Humboldt squid, and in the parasitic vector function performed by freshwater snails. Each of these mechanisms has evolved independently, shaped by particular selective pressures in different ecosystems, which makes the phylum Mollusca a natural laboratory of convergent solutions to the fundamental problems of predation, defense, and reproduction. The saxitoxin of mussels, the tetrodotoxin of the blue-ringed octopus, the conotoxins of the cone snail, and ochidaic acid of contaminated bivalves are chemically distinct compounds produced by different biochemical pathways and with mechanisms of action that differ in their molecular details, but all converge in producing neuromuscular dysfunction or tissue damage in vertebrates. This chemical convergence toward the same functional target, the nervous system of vertebrate predators, reflects the universality of nervous signaling systems as a point of vulnerability in animal biochemistry, and the ability of evolution to independently discover the same solutions when selective pressures are equivalent. Parasites that use mollusks as intermediate hosts add a dimension of indirect danger that escapes intuitive perception, but that, in terms of years of life lost and disease burden, far exceeds the combined impact of all direct poisonings by venom. Schistosomiasis alone represents, according to the most conservative estimates, more than 3 million disability-adjusted life years lost annually, a figure that puts into perspective the statistics of deaths from stings of Conus geographus or from bites of blue-ringed octopuses, which are counted in dozens annually. The component of mystery does not disappear when compiling this inventory of known dangers. On the contrary, it becomes more evident. Most of the known mollusk species have not been studied toxicologically in a systematic way. The deep ocean floors harbor species that have barely been photographed, and of which no living specimen has been examined under controlled conditions. The interactions between mollusks, their symbiotic bacteria, and toxin circuits in marine food chains are incompletely known. The mechanisms by which some bivalves accumulate certain toxins while avoiding others, and the possible adaptive functions of that selective behavior, are being actively investigated without yet producing a coherent picture. What the phylum Mollusca demonstrates with this diversity of lethal and dangerous strategies is that evolution has no aesthetic or functional preferences. It can construct a pharmacologically precise venom in a snail that fits in the palm of the hand, or produce a 200-kilogram predator with armed tentacles in the Antarctic abyss, or transform a small freshwater snail into the vector of one of the most devastating infectious diseases on the planet. Each of these solutions is, from the point of view of evolutionary biology, a resounding success, accumulated over 500 million years of history in the oceans, rivers, and terrestrial land of a planet that has not been at all hostile to these animals.